This is Professor Nandita Bhakat from Department of Botany, Narajole Raj College. Today in this session, we will be discussing about operols. So, what are operols? We find these operols in the prokaryotic cells. Here we find that many different genes, they are involved in only one function, giving result to a single function. According to the new gene concept, what we know is that one gene is responsible for one function. This is a modern gene concept. But in the case of E. coli, we find some exception. What is that? In the case of E. coli, we find that many different genes in different loci, different genes from different loci, they are working together to give or to result a single metabolic function. Okay, so that is all about the operon, the lac operon, that is the lactose operon. We will be discussing about today. And glucose is the ready-made energy source for we are studying E. coli over here, bacteria. In the presence of glucose, E. coli is automatically taking the glucose from the medium and the operon is off. But when glucose is not there in the medium and lactose is still present in the medium, then what is happening? Then the operon is turning on. Okay. So before describing it in details, we have to draw the structure of the operons, and then I will discuss it part by part. Okay. So this is the basic structure. So this is the promoter for regulator. This is the promoter. This is a regulator. This P stands for promoter. O stands for operator. This is the operon. From this promoter, operator and the structural genes, this is the operon. and these are the structural genes. This LAC-Z, LAC-Y and LAC-A, these genes, they code for different enzymes having different functions. So this LAC-Z, it encodes beta galactosidase. LAC-Y, beta galactoside permease. And this LAC-A, beta galactoside transacetylase. Okay. So these are the enzymes encoded by these three genes. The LAC genes provide an example of signal integration. Their expression is controlled by two signals, each of which is communicated to the genes via a step regulator, the LAC repressor and CAP, cyclic adenine phosphate, CAP respectively. The LAC operon of E. coli is one of the two systems French biologist Francis Jacob and Jacques Monod in formulating the idea about gene regulation. It has two regions, the regulatory unit region and the structural unit. These are the regulatory region which regulates the structural genes, and the other three are the structural gene regions. The regulatory region comprises of the inhibitor. This regulator is also known as inhibitor, and this is a promoter for inhibitor. So this inhibitor or the regulator, then the promoter, operator, these are all the regulatory regions, and the structural regions are the LAC-Z, LAC-Y and LAC-A. So there are two regions, one is regulatory part, the regulatory genes and the structural genes. We know that the LAC-Z is encoding the enzyme beta galactosidase, which cleaves the sugar lactose into galactose and glucose. So galactosidase cleaves the lactose into galactose and glucose, both of which are used in the cell as energy sources. LAC-Y, which encodes the protein transferase, a protein that inserts into the cell membrane and transports lactose into the cell. It increases the cell permeability, it increases cell permeability so the lactose can enter into the cell. LAC-A gene encodes thiogalactoside or beta galactoside transacetylase, which rids the cell of toxic thiogalactosides and also gets transported in by LAC-Y. So now this is the basic structure and the functions of the different structural genes for which lactose is inserted into the cell. It's broken down into galactose and glucose. Now we will study about the changes or the functions which occurs when glucose is present in the medium. So hopefully this is clear with you. Now I am going with the presence of glucose in the medium. At that time, how these genes they function when glucose is already present in the medium. So let me write over here when glucose is present in the medium. Remember, when glucose is present, that means the operon need not to be start on. It will remain off. In this operator region, so this inhibitor, when glucose is present in the medium, this need not to be function, so the operon needs to be switched off. What will happen? This lac or the inhibitor will produce the mRNA. From here we will get the protein, that is the repressor protein. Repressor protein. This repressor protein will bind with the operator over here. Okay, so repressor protein is binding with the operator region. RNA polymerase, it will bind at the promoter. Okay, this is RNA polymerase. It binds at the promoter region. Now, as because the repressor is present here, the RNA polymerase cannot move over these structural genes to produce enzymes, and hence no enzymes will be produced from the structural genes, and hence the operon will remain stopped. This repressor, it inhibits further transcription. Okay, so what is happening now? This is prohibiting RNA polymerase to transcribe these structural genes. This is the case when glucose is present in the medium. Glucose is the preferred energy source, and hence when glucose is present in the medium, the lac operon remains switched off. Two regulatory proteins are involved. One is the activator called the cap, and the repressor called the lag repressor. The lag repressor is encoded by the lag I gene, or this gene, which is located near the other lag genes but transcribed from its own promoter. This has its own promoter. The name, this cap stands for catabolite activator protein. Catabolite activator protein. CAP stands for catabolite activator protein. The gene encoding CAP is located elsewhere in the bacterial chromosome, not linked with the lag genes or the lag operon. Both CAP and lag repressor are DNA binding proteins, and each bind to a specific site on DNA at or near the lag promoter. The binding of both the cap and the lag repressor on the DNA prevents RNA polymerase binding at the site, and hence transcription is inhibited. As I told you, binding of this repressor over here inhibits the RNA polymerase to move further on, and hence transcription is switched off. Now we will come to the opposite thing, or the other thing, that is when Instead of glucose, lactose will be present in the medium. Let us see what happens over there. Hope this is clear with you. Now when lactose is present in the medium, this is the promoter of this inhibitor gene. Okay. So from this will it's better to draw from here than from the inhibitor. We are getting the mRNA, means this gene is transcribing and we are getting the mRNA, and after translation we are getting the repressor protein. It's a repressor protein, I am adding it RP. Now when lactose is present in the medium, lactose is there in the medium. When this lactose is coming in association with this repressor protein, in the structural change we can find the repressor protein. The structural change we can find. And as because this, it is not going to bind it on the operator. It will not bind in the operator region, and hence, as usual, the mRNA will bind to the promoter, and hence, as because no obstruction is present in the operator region, so it will smoothly go and transcribe all the structural genes. And as its transcription goes on, the different enzymes will be produced from the particular genes. When lactose enters the cell, it is converted into allolactose. Okay, when lactose enters into the cell, it is converted into allolactose. What is the basic difference between lactose and allolactose is that in lactose, glucose and galactose, it is binded by beta 1,4 linkage. Beta 1,4 linkage. And here glucose and galactose, they are bound with this 1,6 linkage. This is the beta 1,6 linkage. This is the basic difference between allolactose and lactose. So it is the allolactose that controls the lac repressor. Paradoxically, the conversion of this lactose into allolactose is catalyzed by beta galactosidase enzyme, itself encoded by one of the lac genes. Allolactose or lactose binds to the lac repressor and triggers a change in the shape of the protein. So this allolactose, it binds with the repressor proteins and it triggers a change in the shape. And for this change shape, this is not going to bind with the operator. As a result, mRNA is free to move and transcribe all the structural genes. In the absence of allolactose, when the allolactose is not there, then what will happen? The repressor in its original structure will bind to the operator and will prohibit or switch off the transcription. Once the allolactose has altered the shape of the repressor, the protein can no longer bind the DNA, and so the lag genes are no longer repressed. What we can see over here. CAP activity is regulated by a similar manner. Glucose, it lowers the intercellular concentration of a small molecule, CAM. This molecule is the allosteric effect of CAP. Only when CAP is complexed with CMP, the protein adopts a conformation that binds DNA, and also when the glucose level is low and CAP binds DNA and activates the lag genes. So this is what we have studied today. When lactose is present. Then this situation occurs as because there is no restriction over here, so the mRNA can move and transcribe all the genes, and after these genes are transcribed, lactose can enter into the cell and is broken down into glucose and galactose, and from that glucose, the bacteria, it can means that glucose acts as a source of energy for the bacteria. Okay. Thank you.